Morning. Welcome to Video 16, Unit 1.3 for Mr. McGee's 9th grade biology course at Arizona School for the Arts. In this video, we'll be taking a look at the diversity of cells and an idea called endosymbiosis theory, which tries to explain the origin of eukaryotes. This diagram shows us how every living thing on Earth is related. They all share a common ancestor. The common ancestor is the little line at the bottom in the center of this diagram. And even though we don't know who that was or what that particular cell may have looked like, we do know that over the course of about 4 billion years, living things have diversified into around 30 million species. Maybe more, maybe less, but that's a big number. It turns out all of those living things fit on 3 major branches: bacteria, archaea, and eukaryota. These are called domains because they are such huge branches of the family tree of living things. 2 of these domains, bacteria and archaea, are single-celled organisms that have no nucleus. And so a lot of times scientists or biology teachers will lump them into a group together. They'll call them maybe quote-unquote bacteria because they sort of look the same superficially, or we might call them prokaryotes because they don't have a nucleus. But you should know that bacteria and archaea are really very different organisms. They have different biochemistry, different types of metabolism. They make different proteins. Genetically, they're very different. So they're really 2 large branches that separated a long, long time ago. In fact, some scientists think that the archaeans are more closely related to eukaryotes than they are to bacteria, even though superficially they look the same. Everybody else fits in this group, the eukaryota, and these are organisms that you may be more familiar with, things that are multicellular, things that you can actually see, right? Like plants or animals or fungi or even Hector. So the group prokaryotes, that's not a real taxonomical group, meaning that it's not a group that shares one direct common ancestor, but it's made of 2 major groups. It includes the bacteria. Sometimes that's called the eubacteria. That just means the true bacteria. Or the archaea, which is oftentimes called the archaebacteria. Now what all these guys have in common is that they're all small single-celled organisms with no nucleus and no other membrane-bound organelles inside the cell. So they don't have a complex cellular structure. They're very simple and very efficient. Some of them are colonial, or occasionally they live as large visible clusters, but for the most part they can get along just fine on their own. This is a diagram that tries to show you the basic or more general structures that are found in prokaryotic cells. I don't want you to memorize this just yet. We're going to work on that sort of thing in class. But I do want to I do want you to notice how simple this cell structure is. There's not a whole lot going on, right? There's not tons of elaborate structures. It's very efficient. And because it's so efficient, these guys are able to reproduce quickly. They are resilient in changing environments, and they are able to really become the most popular organisms on the planet. They're everywhere. They do all sorts of different things. There's lots of diversity. They do come in 3 common shapes. There are more cell shapes out there, but we're going to focus on just these 3. So starting in the purple is spherical shaped cells, and when you see round cells like this, the description for that, the term to use is to describe them as coxy. One cell would be a coccus, and a lot of times you hear that reflected in the names, like pneumococcus is a type of bacteria that causes pneumonia. It's spherical shaped cells. Other cell shapes include bacilli, which is in yellow. Those are rod-shaped. So that's sort of like when someone says a bacteria, this is the sort of cell shape that people typically think of, little rod-shaped cells. And then the spiral-shaped cell in orange, cells of this type are called spirochetes. And a lot of times, again, you find these cell shapes reflected in the names of the individual bacteria. The other major group, eukaryotes, is one branch of the family tree. All eukaryotes share a common ancestor. Eukaryotes have cells that include a nucleus, and also inside their cells are cell structures that are enclosed in their own membranes. Now that's important, as we're going to see in a few minutes. So really important cell structures like the mitochondria or the nucleus, those are enclosed in a membrane. And just take a look at what's going on with these cells. This is a diagram that's supposed to be a typical animal cell. Notice how much more there is in it. It's crazy in there. There's all kinds of different structures doing all sorts of elaborate and complex things. And not only that, but it's many times the size of a typical bacteria. So these are really big, complex cells. Now, if we look at cell structures inside of eukaryotic cells, we find something interesting. So the graphic here on the right side is a scanning electron microscope image of a pretty common cell structure called a mitochondria. And when we look at it under that kind of magnification, we find that inside of the cell are folds and compartments of membrane that are very similar to certain types of bacteria that are living on their own and still alive today. And what's even weirder is that mitochondria have their own DNA, and they have their own sort of life cycle. They divide on their own, separately from the cells that they're inside of. If we look at the DNA of mitochondria, we find genes on it, and those genes aren't similar to the genes that are found in the nucleus of a eukaryote, but those genes are similar to the genes that are found in other bacteria, in living bacteria. That's really weird because it makes it seem like the mitochondria was once a free living bacteria that now is part of a eukaryotic cell. So we think that this is how eukaryotes evolved. We think it started with one ancestral eukaryote that did not have a nucleus or any other cell structures. And we think it happened in at least 3 steps. First, we think that the cells grew really large, and being big cells makes them sort of inefficient. They need to be able to transport nutrients from outside the cell deep inside the cell, and in order to do that efficiently, these cells developed infoldings of the plasma membrane. So the cell membrane folded in to make channels and tunnels. Once that happened, we think that infolding of the plasma membrane became the nuclear envelope, which helped to protect and package the nucleus and the genetic material inside, and then the endoplasmic reticulum, which is like a network of tunnels and passages that helps transport things. Now, eukaryotic cells are really good at breaking down sugar into 2 simple components, and the details of that we'll talk about later, but they can break down sugar to some degree, but not very efficiently. We think that the next step involved developing a symbiotic relationship with bacteria that could help to break down sugars more efficiently. And those bacteria moved into the cell, helped to break down sugar, helped the big eukaryotic cell survive, but also were protected and given nutrients by the big eukaryotic cell. And eventually, over long periods of time, those bacteria just became permanent residents of the cell. They became the structures that we know as mitochondria. Now that branch became all of the heterotrophic eukaryotes, so all the eukaryotes that can't make their own food. A third event may have happened after that, which involved engulfing photosynthetic prokaryotes. That means photosynthetic bacteria came to live inside eukaryotic cells and produced sugar, which the cell could then use for food. And maybe the cell provided important resources like nitrogen or phosphorus to those photosynthetic prokaryotes, resources that are hard to get in an environment. But either way, that branch of the eukaryotic family tree, we think, became all of the photosynthetic eukaryotes. Now, the more that we look into this, the more evidence we have for multiple events of endosymbiosis leading to cell structures in eukaryotes. This diagram tries to show us how many different times that may have happened in the history of eukaryotes. We think at least 5 major symbiotic events happened in the evolution of eukaryotes. But I know what you're thinking. Where do we find examples of this process happening today? And one of these examples is found inside the gut of this animal. If you remember, termites eat wood. Wood is made of cellulose, and cellulose is indigestible by animals. So this guy has to have some symbiotic organism living in its gut to help break down the wood. And it has that. Here's one of them. It's called Mixotricha paradoxa, and it's not a bacteria, it's a eukaryote. So it's a big cell, and it swims around inside its gut, breaking down chunks of wood into simple sugars. The problem is that Mixotricha doesn't seem to have any of the genes to produce enzymes to break down cellulose. So how is it doing anything to benefit the bacteria? Well, if you look at the red circles, what you find is tiny little symbiotic bacteria that are living embedded in the cell membrane of Mixotricha paradoxa. The little fuzzy ones over on the right, those may look like cilia, but they're actually spirochete bacteria that are embedded in the cell membrane and live there, and Mixotricha paradoxa provides them with food, and then as a result, they respond to stimulus by Mixotricha paradoxa, and they'll wiggle, and that allows Mixotricha paradoxa to swim around. Now on the other side, on the left side of this slide, the other red circle is indicating tiny colonies of bacteria that actually break down cellulose. Those live on the nose of Mixotricha paradoxa, and so as it's swimming around through the gut of the termite, those bacteria are encountering the particles of wood, they're breaking them down into glucose, Mixotricha is absorbing the glucose, and then inside Mixotricha is at least 3 other symbiotic bacteria that are helping to further break down those sugars. So inside Mixotricha bacteria paradoxa, or living embedded in its cell membrane, are at least 5 organisms. That's bizarre. Well, that's the end of this video. Please complete the corresponding review questions and be prepared to show me your notes in class. Oh, and here's your bunny of the day.